Now, in the previous tutorial, we looked at the databases where you can see the protein that you wish to express. It will tell you the size, it will tell you how many amino acids, and you decide whether it's suitable. So also if it's like the location in the cell will influence whether it's going to be suitable for expression or the level of difficulty. And so in this one, we're going to look at where you then get the nucleotide sequences, because if you want to express it, it's the nucleotide sequences, specifically the coding sequences that you want for placing into a plasmid vector. For expression, initially all the vectors, you're going to start with plasmids, and then if you want to use a viral expression, that viral proteins that are required will then be expressed alongside with the plasmid. And so we always starting with the plasmid. So let's look at the databases where you can get that. So several databases that you can get that from are NCBI, there's GenBank, there is UniProt, and there is PlasmoDB. These websites will allow you to get the sequences that you need, but once you have them, then downloading them is going to be quite specific. So if you are in either of these databases, you generally can get a link to go to from one to the other. You'll be able to see links to either the nucleotide sequences on EMBO or on GenBank or PlasmoDB. They try to link with each other. It's useful because sometimes they, like the UniProt, will just have sequences for proteins and they may not have the nucleotide sequences. But if you actually click on its equivalent entry in PlasmoDB, you may find the nucleotide sequences. So don't give up if you don't find it from one website. Check all the other links to see who's got the repository. Another thing to be aware of is sometimes the default download buttons on a database may not work, and this may just mean that you need to find other ways to download it. So for instance, when I went into PlasmoDB, on my desktop, I had difficulty downloading the sequences, but on my mobile, it was fine. And also when I used a different way, so I went to Tools and then Sequence Retrieval. And then there you can specify what you want to download, where you can indicate that you want to download, for example, the FASTA. Then you can specify the sequence type. So here you have either the unspliced genomic region, or you want to find the spliced genomic region, that's your transcribed sequences, or maybe the DNA component, or a transcript component, or protein sequence, or even protein features. So here there are 4 types of sequences that you can access. You have the protein, there's the predicted translation product of the gene. You have the CDS, which is the coding sequence. So this excludes untranslated regions. Introns have been spliced out. Then you have the transcript, which is the processed transcript, including the untranslated regions, but introns again have been spliced out. And then you have the option of downloading genomic sequence, which is a region of the genome. The genomic sequence is always returned from your 5' to 3' on the proper strand. So once you have the sequence that represents your protein, you want to design primers so that you can use the primers to amplify out, to make copies of the protein from its native environment, so from a cell line. But if you have tissue, then you can homogenize the tissue and then use the extracted DNA in order to be able to use the primers to amplify out the gene that you want. If you don't have access to the cell line or even a tissue from which you can then extract or blood from which you can extract the gene representing the protein, then you can have companies synthesize the whole protein for you along with the primers. I mean, people are routinely having companies synthesize the oligos, the primers that they'll use to amplify out a gene representing a protein. But if you have no way of actually getting that protein, that gene representing the protein from a cell line or from tissue, then the companies can also make the whole gene, synthesize the whole gene for you. Mind you, it's going to be very expensive because instead of the 21 or so nucleotides or 18 base pairs that you would have synthesized as an oligo, they now have to synthesize a whole gene representing a protein. So with that, we come to the end of this video. I hope that it was helpful to you, and if it was, please give it a thumbs up. If you're not a subscriber, please join the study group, and I'm going to catch you in the next video, where we'll be looking at vectors, the type of vectors we can use to express the genes that we amplify out of its endogenous or native environment, or if we had a company synthesize it. We're going to use that vectors that can take those genes and allow us to be able to express it in a different cell line, in a different host, or maybe the same host, but the specific genes that we want.